So welcome here, everyone. I'm Erik Söderbäck. I work for Covaris, and we will talk about FFPE in oncology settings today. So we have an agenda. I will go through what Covaris can provide in terms of sample preparation and also library preparation. We will do an introduction. So Nadia will do an introduction from Illumina. And then we'll show a collaborative effort from Covaris and Illumina where we optimize the workflow for Phi-base, methylation, and mutation sequencing. And it's a streamlined solution for FFPE analysis. And then we'll end up with Professor Georgi Markovarga from Lund talking about the proteomics workflow from FFPE material. So he will describe then how to prepare protein and in a mass spectrometry workflow. So Covaris, probably most of you know Covaris as the DNA shearing company, but the technology we base our solutions on is called Adaptive Focused Acoustics, and that can be used for much more than just the shearing of DNA. So it's a disruption, so it can be used for disruption, cell lysis, molecular processing, and the trick is that we transfer very accurately the energy to the sample. And it's a non-contact isothermal process which eliminates any cross-contamination and it preserves the analyte integrity as well. And for this we have the consumables, accessories and reagents that are optimized for the application of interest. And we can say that you can work with more or less any kind of sample types and with any kind of biological molecule. So our offerings can be summarized like this, that we have instruments from single sample handling up to high throughput, 96 and 384 well systems. And for this we have then the consumables and the reagents that are required for certain applications. And we can automate this as well. And I'll come back to the automation regarding FFPE. And for this, we serve more or less any market that uses biological molecules. And we can also support applications for any kind of omics. So we have solutions for genomics, transcriptomics, methylomics, or methylation analysis. We can also do for metabolomics. But going into the truXTRAC, which is our reagents then for extraction of DNA and RNA from FFPE, this is quite a simple workflow, and we use the ultrasonication device then to emulsify paraffin, which is a big difference then from other vendors that have organic solvents for the deparaffinization. So in the step here, we use the alpha technology and we emulsify the paraffin, which gets small pieces of the paraffin and the tissue out in an aqueous-buffer. And by that, we can efficiate the proteinase K treatment downstream. We spin down the sample, which makes the DNA ending up in the pellet and the RNA in the supernatant. And by purification of both these fractions, then we have DNA and RNA from the same sample, which can be used for further downstream processing. And the quality is very good, both for DNA and RNA, and particularly the RNA sticks out as very high quality with a high molecular weight RNA. But to show that also the DNA is of high quality, I want to emphasize an application note recently from PacBio, which show that with DNA from FFPE, you can do long-range sequencing. And with amplification library preparation, you can reach very long reads then. And they claim that these results demonstrate that even with degraded FFPE material, it is possible to recover DNA fragments of sufficient quality and length to enable high-fi long-read sequencing. For short-read sequencing, Covaris has during the last year introduced library preparation solutions for whole genome sequencing as well as for RNA sequencing. These can be used for any material, but we have optimized workflows for FFPE DNA as well as from FFPE RNA. And now, latest here, we have also the multi-omic library prep with Illumina 5 base, and that's what we're going to focus on now. So I leave this to Nadia.
Thank you so much, Erik, for the opportunity to tell you a little bit more about the 5-base solution and also our collaboration with Covaris. We've been partnering already for a long time, also from the time of the TSO 500 assay, and it's very nice to see that we're taking it also to the next level with the expanding range of applications. So, and we jointly, we believe in the power of the multi-omics approach as the way to reveal, more complex biology and to see the fuller landscape of what, and to ask the questions that the cancer biology really deserves. So with this in mind, we've been from Illumina side active on the sides of the very different omes. Nowadays, many of them can be addressed with the next generation sequencing. And thinking about DNA in general, it's, it's in the DNA's biology to be a multi-omic molecule because it brings two levels of information at the level of genomics, so with all the variants that one might be interested in, but also the additional level of regulation then epigenetics brings in particular methylation. And this is exactly what we are trying to address with the Illumina 5 base solution. The innovation bit that we are bringing here, Illumina is earlier, just it's been still kind of a recent announcement from the last year. We brought it to the market. This novel type of the library preparation that allows you in one go, in one workflow, without the need of the splitting things in two, get the information both from the genome level, right, in terms of when you're looking for the variants, but also at the same time getting the full picture of the methylation profile. And, I would not go into too many details, but the essential thing, how it works, the first step is bringing the innovation on the side of the chemistry. So on the left side here, you see the traditional NGS-based methods or bisulfite sequencing, when you would need to treat the molecule of interest with the enzymes doing the non-direct conversion. So everything that had the methylation will be sort of protected, and then, all the non-methylated Cs would be converted into Ts. As a result of this, the methylation component was there, but the library complexity was quite, shifted, because essentially you are ending up with the overrepresentation of Ts. And also this has been quite a harsh treatment that does not preserve the integrity of the molecule. So the approach that Illumina take in bringing their solution to life is really modifying the way that the chemistry works. And this is what we are looking at the right side of the slide. Illumina 5-based conversion chemistry enables this direct conversion of the methylated Cs into Ts, and that brings a whole number of advantages that I'm going to highlight just on the next slide. So by doing so, we're essentially addressing the gaps in traditional methylation sequencing methodologies. As I mentioned in the beginning, the assay is made to bring you the dual insights, so the high accuracy detection of both methylation and genomic variants. It comes with the advantage of the seamless workflow, and again, with the partnership with Covaris, we can really take it from the step of the extraction all the way to the data output, and it drives the cost down significantly. I would kind of, like to also give you a snapshot about the workflow, what it takes from the kind of, library preparation into getting the insights. So the library preparation step takes less than 1 day. And then we are speaking about, the sequencing part that is also can be done on the range of Illumina instruments, and we are wrapping this part up with a very important bit. It's the power of the software, because again, as I always like to quote, it's software that brings the data, that makes the data the insight. So this is the overview how it works, and the data analysis part is enabled by the Dragon pipeline being the part of this integrated solution, so you don't really need to bring the additional bits and pieces. And you know, as our collaboration with Covaris, we started, you know, we started sort of with the first testing on the workhorse, right, of our customers with the LE 220, but then we expanded basically this workflow, the testing of the workflow, to the other platforms as well. And Erik, I will invite you to stand next to me to comment on the outcome.
Sure, the instruments you see here, so the workhorse called here, the LE220 is present in a lot of labs around the world. But during the last years, we've introduced a high throughput solution called R230 and also one medium throughput ML230, which both works in 8 strips, but also the R230 can work in 96 well format. And it can also be automated, and I'll come back to that. But in principle, what we showed here with genomic DNA, the Covaris, the instrument, supports the 5-base workflows, irrespective of the throughput then. So we have scalable solutions then for the 5-base. And the Illumina 5-base and Covaris then consistently deliver a high confidence variant calling and methylation profiling. This was from genomic DNA, and then we took the next step then to optimize also workflow for FFPE. So here we took the genomic DNA from a reference DNA, and we shared that down to 200 base pairs to have it equivalent to what we get from the FFPE, and we compare the outcome then from the library preparation and sequencing. And just looking what the cell cultures that are then formalin fixed and embedded in paraffin, and we extract that with our truXTRAC solution and run through the 5-base workflow. You can see here it's very consistent then with the library traces you see to the left, and also the amount of library that we get out from this. So irrespective if it's genomic DNA or FFPE DNA, we can provide the workflows. And also the QC metrics from the sequencing shows very robust data from the FFPE as well. So here it shows that we get more than 200 base pair on average insert size, and it's no impact on mapping or the methylation analysis. It's equivalent between the genomic DNA and FFPE DNA. We also investigated the variant calling from this, so both the SNPs, single nucleotide variants, and indels show very high consistency between the genomic DNA and the FFPE DNA. So from this we took it to also, of course, to look at real patient samples. So the last step then, we took lung cancer and compared to lung normal tissue. And we extracted this with truXTRAC and run it through our R230 platform for both the extraction and the library the DNA shearing and library preparation. And again, so to the left, you can see the size distribution then of intact, the FFPE DNA, which is very intact then. And it's quite high. It's distorted then with the x-axis here, but the peak range here is about 5,000 base pairs from FFPE DNA. And when we share this for the optimized workflow for the library preparation with 5-base, it's about 200 base pair, and that's the trace you see to the right, that you get very consistent library, the fragment size for the library preparation. And to sequence this, again, we show that the quality metrics are very equivalent here. So for both the lung cancer tissue and the normal tissue, we show consistency for the coverage, the mapping, but also the insert size around 200 base pairs. So to summarize the results from this, we show that both we have a scalable solution for the instruments up to very high throughput. We can show consistency with the methylation we show here. It's consistent with the positive and the negative controls. both input-wise, so we compared here 50 nanogram and 100 nanogram. We compare genomic DNA with FFPE DNA, and the results are very consistent both between genomic DNA and FFPE DNA. So to summarize this, I give Nadia the way to describe it from the Illumina standpoint.
Thank you so much. So today what we try to show you is also the way how together with Covaris we did really go in a nice direction in streamlining the genomic and methylation profiling for FFP material, providing this comprehensive solution that really can allow you to do more samples, to do them faster and then accelerate the research workflow, starting all the way from the nucleic acid extraction, also with the truXTRAC prep from Covaris, going into the DNA sharing that is here across different platforms, so depending also on the throughput of your team and the accessibility of the instrument. This is a really great point. And then seamlessly going into the library preparation, one library prep, so no additional kind of workflows, sequencing as an outcome, you get the information like 2-in-1, you get the information about the DNA sequence and the full methylation profiling. So this is exciting. We've been now partnering, of course, on establishing, testing these methods, and we are working on the preparation of the joint technical note, so this will be also available very soon together with the other resources that are out there already. Thank you so much.
Thank you, Nadia.